Okay, this artificial hybridization is an important process in plant breeding programs. Plant breeding programs involves production of better progenies from the superior plants. Here I am using a word superior. Why superior? Because the plants that we are going to cross with each other, they are having the desired features that we want to have in their progenies. So, in plant breeding, a very important process, artificial hybridization, is performed so that the desired characters of two parents can be mixed or combined and can be seen in the progenies of those plants that we have crossed. Okay, so what are different steps of artificial hybridization? Why I am saying artificial? Because this is not a natural process. Here the hybridization means crossing between two parents of different genotypes, different genes. They are crossed by the human beings, or this is not a natural process, it is performed by the intervention of human beings. Okay, so artificial hybridization is actually focused on enhancing or improvement in quality of given plants. For this, we have to firstly select what type of plant we are going to cross. Say for an example, we have two plants A and B. We have selected them for the crossing purpose. Say it is a sugarcane plant. It is having a thicker stem, but the length is less. While the B plant is also a sugarcane plant. Its stems are very long, but the thickness is very less. And in case of sugarcane, what we want? The length should be very high and the stem should be thick so that the production is increased. So to increase the production, we need such a progeny which will be having both the combined features. Means the progeny should be having thick stems and more length of their stems. So for this, we can have artificial hybridization between these two parents so that the progeny can have the desired feature of both of these parents. So when we have selected the parents, now the step will be the artificial hybridization. How we are going to perform this? For this, I have drawn here male and female parts of spore flower of these plants, any plant that we are going to take in our experiment. This is the pistil part, the female part, and this is the stamen part. And here you can see that this flower, if it is bearing both pistil and the stamen part, it is a bisexual plant or bisexual flower. This is a bisexual flower. Now we don't want that the pollen grains from this flower should fall on this stigma, because we don't want to cross, we don't want that the egg in this pistil should fertilize with the male gamete of the same plant, because we want to introduce new features. So, for example, this is the flower of this plant. Then what we have to do here, we have to remove these stamens or to make them a unisexual plant, unisexual flower. So the first step of artificial hybridization will involve the emasculation. Emasculation. This is very important step, emasculation. When we have selected the plant, then we will emasculate the flower. What we mean by emasculation? It requirement to remove any the given part, because it is having only it will be having either the stamen or it will be having the pistil only. So emasculation is not required in unisexual flowers, but here we will make this bisexual flowers. They are emasculated, means their anther part is in the young stage. Young stage means if the microspores they haven't formed, the anther is still young, and sporogenesis, microsporogenesis process hasn't started. Then in that condition we are going to remove the anther before the dehiscence of anther, before the formation of microspores. Otherwise, if it dehisces, then they can fall over the stigma, and our artificial program will be it is not going to be a successful one. So we have to emasculate the flower. Okay. Now the next step is bagging. After emasculation, we have to bag the flower. Suppose I have removed the stamens here. I have emasculated the stamens here. One more thing I have to make here clear, that if the stamens are very long or the flower is bigger in size, then it is very easy to remove the anther by cutting them. You can have scissor and we can cut them. We can remove them by plucking them, or we can cut the tip of the stamen, we can remove the anther. But if in case the flower is smaller in size, it is not easily removed by manual process. So in that case, we can have a suction pump also. So in some cases, if the flower is smaller, then the suction pump can be used to suck the anthers from that small flower. Or if in some cases the length of stamen is very long, so this length is very long, then in that case we can also dip them in boiling ethanol so that the tissue can be damaged. There are different methods to emasculate the flower. Okay. After emasculation, what I was telling you, that the process is, the next step is bagging. So firstly I am going to remove this anther and the stamen, you can say, and this is the emasculated flower, bisexual flower that has been emasculated. Now we have to cover this pistil or this female part with a butter paper bag, or the polythene bag. Why we are covering the structure with a bag? Because we have removed the anther or stamen from this flower, so autogamy is not going to take place. But we can't control the pollination through various agencies from other sources. This structure is still vulnerable to pollination with the other sources, like it can be geitonogamy or it can be from other plant that is growing nearby. Okay, so to prevent the cross pollination, natural cross pollination, we have to cover this structure so that undesired, suppose again give an example, suppose another flower of the plant, same species plant, can dehisces and its anther, and this they anther after dehisces, the pollen grains they can land over the stigma of this flower that we are not desiring. Okay, so to prevent the natural pollination process, we are covering this structure with the bag. So this step is bagging. Third step is pollination. Pollination here will be the artificial pollination. Why I am saying artificial? Because we want to cross plant A and B. Suppose this is the flower of emasculated flower of plant A. Now we have to dust the pollen grains from that plant that we want to cross with this A plant. So we have to take up the pollens from the B plant. When the dehiscence will take place, then we will collect the pollens from that plant B and will dust it over the stigma surface of A plant. These pollens that we are dusting, they are the pollen. Dust the pollens from the bee plant, this bee parent. The bee parent will be having its intact anthers. They are undergoing the microsporogenesis process, and these anthers will be also covered. When we have covered this plant with bag, these anthers are also covered with bag, so that when dehiscence will take place, the pollens are collected in the bag. Okay. Now, after dehiscence, we have collected the pollens in that bag. Then we can dust those collected pollens from this bag. The pollens are from bee plant. They will be dusted over the surface of stigma of A plant. So this pollination is under our control. We have pollinated this flower with the desired pollens that we want. We have prevented the natural pollination by bagging both of these structures. Here we have covered it with covered the pistil with bag, and here we have covered the stamen or anther with bag, so that any undesired pollen grain should not reach on this surface or should not mix with these pollen grains. Okay. After pollination process, the next step will be rebagging. Why rebagging? Because means again. Rebagging is required. Firstly, we have removed the bag, dusted the pollen grains here, and then again we have to cover this pistil with the bag again. Rebagging is required because the pollination from natural agents can't be controlled if we haven't bagged it again. We have again covered it so that any undesired pollen shouldn't land on this stigma, shouldn't mix with the pollens that we have dusted over its surface. And after pollination, the pollen germination will take place, and then ovule that is having the egg inside it, fertilization process will proceed, and fruit formation and seed formation will take place. This is how, if you want to cross two selected plants with each other so that we can combine their feature, so that we can improve the variety of plant, enhance its quality, we have to follow the artificial hybridization.